So first of all, welcome, everyone. Today we're going to talk about something that started as a journey probably 4 years ago. I'm looking back at Philip Payne nodding his head in the back of the room. This was a long and sometimes arduous journey to get to this point, but we are finally here at the point where the Center for Translational Bioinformatics has been established here at Washington University. And today I'm going to talk a lot about, I'm going to keep things at a fairly high level. I am going to show some examples of things that we're already starting to do, have already done, and are translating into some of the things that the center is going to make available to researchers across the campus. I am going to keep things at a relatively high level just because in a 45-ish minute talk, obviously we can't go into a tremendous amount of detail, but we will obviously have additional seminars in the future and obviously be willing to talk with folks in the future. So this center has actually been established within 2 different institutes. So this is a somewhat unusual thing, but in this case it made a lot of sense. So the Center for Translational Bioinformatics exists jointly in both the Institute for Informatics, Data Sciences, and Biostatistics and the McDonald Genome Institute that you see up here. And really the job of this center is to very much bridge what those 2 institutes do and what a lot of folks within this institution do that go across the research and clinical boundaries. And that's really what I'm going to talk about today, about the mission of this organization, a little bit about how we're structured, and mostly about how we're going to do what we're going to do, and show a few little vignettes of how we're building on prior successes to do this. So what is the CTBI? I kind of mentioned a little bit. I'm going to go through sort of the mission and the goals in a little bit more detail. I'm going to talk about our scope of what we're going to be doing. I'm going to just briefly talk a little bit about the early structure of the organization. Then I'm going to spend a lot of time talking about how we're going to achieve those goals, because when you see the goals, they are not small goals. They are rather audacious, frankly, in terms of what we're trying to do. And then we will kind of finish up with just some logistical stuff in terms of summary and some acknowledgments. And actually, there's going to be a little bit of a plug at the end for another symposium that's coming up in about a month or so. Okay, so the overview of the mission of this center. As I mentioned already, this is the center is jointly housed within the 2 institutes. And our mission is, at the highest level, is to advance the precision medicine paradigm at an institutional level. In order to do that, we have to first recognize the key challenges that make this very difficult, of which there are many, but there are 2 in particular that I'm going to talk about. We're going to utilize highly advanced computational methods to support the work that we're doing and what we're talking about. We're going to leverage a lot of existing expertise. We're not going to go reinvent the wheel. So where there's already expertise within the institution, we're going to leverage that. And we're going to create an environment in which all Washington University investigators and their laboratories can utilize and benefit from what this center is building. So precision medicine is actually a pretty large umbrella, right? There's a lot that goes on in order to do precision medicine. And so in order to attack this, we're actually breaking out the mission of the center into 3 major pillars. So the first is along the lines of what's sort of embedded in our name, and that's translational research. That's going from preclinical to clinical, or from preclinical models to human subjects research. And we are basically going to build a lot of extensive tools that allow this to happen at a much more robust level than is occurring today in a lot of cases. We also want to look at precision therapeutics. So, and what I mean by that is the right treatment for the right individual, right? Getting the right treatment or the right therapeutic to the individual who needs it in a timely fashion is something that you see hints of this in places like oncology, where we test for the different somatic mutations that exist within a tumor, and then we target the therapies around those somatic mutations. But there's a lot of disease areas where we really don't do this. Someone comes in with signs and symptoms. The first thing they do is they go on the cheapest drug that's been around the longest. If that fails, they go on to the next drug. If that fails, they go on to the next drug. That's not really precision medicine. And then the third pillar of this will be healthy aging. So the healthy aging is something we will really start our planning on in the 2nd year, but that's really about how do we understand how to work with people and let them know what things would benefit them in particular. So you hear things about like, well, vitamins do work or they don't work. Well, they work well for some people, they don't work well for other people. Different types of foods and lifestyle, exercise, et cetera, right? And so even with the idea of aging in such a way that we are avoiding disease, it probably needs to be done in a more precision way because everybody's a little different, right? Human diversity is a good thing. It's what keeps us all from being wiped out from a single bug when it comes around, but it also makes the practice of medicine a little bit tougher. I'm also going to talk a little bit about the foundation that we're going to use to build these pillars. So as you would imagine, some of the foundation that we need to build can actually go across these pillars in terms of its use and application. Turn that off. There we go. Okay, so there's an integrated knowledge system that we have already built to some degree, and we're actually advancing that in an artificial intelligence system that we're building on top of that to support many of these pillars. So the requirements that we set out to do this. First, we need to significantly enhance the users, not replace them. That was our first intention. I borrowed this from Philip in one of his slide decks, this idea that the human plus the computer is greater than the human, not necessarily that the computer is greater than the human. I would say that we're actually targeting our corollary, which is the human plus the computer. We hope to be much greater than the human alone or the computer alone. We want to enable both expert and non-expert users. We don't want people who are experts in artificial intelligence or computational biology to be the only people that can use these systems, or clinical informatics. We want basically all the researchers to be able to benefit from this. And we will build these systems to be usable institution-wide. The value-generating components that we create are going to go into place as they're ready. This is not going to be an all or nothing. This is not going to be we're going to go sit in the background for 3 years building some huge monolithic system, and you got to wait 3 years for something to come out. We actually have components of this that may start to be available just within the next month or two. And then we will continually roll things out as we go. As development makes these components available, we are going to make them available to people within the institution. And another component that was really more for the EF is that the initial institutional investment in this center, with that, we expect to become a self-sustaining center as we go forward. So the scope of what we're doing looks a little bit like this. There's a lot of words over here on what would be your left, but really the graph kind of covers it. The idea is that when most people talk about translational research, they're talking about going from preclinical research to human subjects research. That is absolutely within our scope, and you're actually going to see examples of that and how we're building on that right now. The other component of translation, which I'll show some examples of what we're going to be doing in that space, will be going from the research space to the clinical care space and basically bridging that gap. So we have to have a system that's going to be able to work with so almost hypermodal data, clinical data, literature, omics research data, et cetera, right? These are all components of data that are in play in this translational process. This is just a little bit about the structure of the organization. We will have a core and affiliated faculty. In fact, we have one of our first recruited faculty coming in January. We have some affiliated faculty within the institution already. We're targeting 15 core staff. These are machine learning, bioinformatics, API, web application developer individuals that will basically be involved in building a lot of what I'm talking about and working with the folks across the institute in order to be able to use this. Our core faculty will actually have a seat at the table in this process, and these will be people whose work is very much aligned with what the center is doing and often fits into what the center is doing. And we'll basically be publishing exemplar papers and grants along this line. And as you might expect, being part of two different institutes, we will be sharing a good bit of resources across business administration, marketing, communications, bioinformatics, and project management with the MGI and I2DB. Okay, so now that's logistics and kind of the overview of the center itself. What about getting into some of the details of what we're really going to do? So while there are a lot of challenges to doing what I just said, there are two in particular that are very, very substantial hurdles. The first hurdle is really there by design, and that is the fact that clinical and research data tend to be siloed from each other. Only in rare circumstances are they connected, and in rarer circumstances are they really connected in a way anybody other than a PI working on a small project can actually make the association of data that was collected in a research mode back to the EHR. Okay? This basically, when it does happen, there's a connection in an Excel spreadsheet or a small database on a PI's laptop or a computer that allows them to make a connection, but really no one else can. And this is in order to keep the PHI of our patients confidential. So this is a hurdle that we have to overcome. Not get rid of, we have to still keep the confidentiality of the patient's information. So it is a challenge, but we actually have a couple of ideas of how we're going to do this. The second really large challenge behind this is the fact that just the sheer size, complexity, and types of the data involved in these types of research analytics. GTAC alone, which is one core facility in one institution, generates over 3 petabytes of data annually. So start multiplying that across, the institution, all of the core data generating facilities, et cetera. Now, not all generate 3 petabytes a year, but the point is the amount of data being generated are massive, and it's not just one type of data. There are many different types of data generation that are done in the research environment. There's an enormous amount of knowledge already existing in PubMed. Again, people don't want to reinvent the wheel, and somehow we have to tie all that back to the clinical record. So the modality of the data is also quite extensive. So, first challenge first. So I'm a big believer in computer science. In other fields, there's something called divide and conquer paradigm. Divide and conquer paradigm means if you've got a really big complex solution, you don't come up with a really big complex problem. You don't go out and seek to come up with a really big complex solution. You break the big problem down into smaller problems that are much easier to solve, and then you start putting those together into a complete solution. And in fact, most of what we're doing takes that approach. So the first thing that we're looking to do is we realize that we have to build some type of secure data bridge between our clinical and research information for samples that have been consented for clinical use, or for research use, excuse me. And so this is going to be a big deal. We have to figure out how to do this, and maybe even have to figure out how to do this at multiple levels. Because there are things that, yes, we know someone would have to go back to the IRB with a protocol to be approved where they would be able to access PHI. But there are other ways in which we can look at data at a group level out of the EHR that may not require that same type of approval and might be usable by researchers across the institution. And in fact, what you'll see is we're actually targeting both of those solutions with what we're trying to do. So this is what a typical workflow looks like. Someone has consented a sample for research use, and it may go to one of our sample collections, TPC. I'm going to talk about the BioStore in just a bit. Daughter samples of that particular sample may end up at a data generating facility like GTAC or MTAC or any of there's a multitude of data generating facilities at this institution alone. And that sample may end up there, or daughter products of that sample may end up across a number of these, and research analysis is done on that, and except maybe for the PI, if they had consent to do it, that's where it ends. There's no going back to the clinical record anymore, right? And what makes it worse is that a lot of these groups, and I will admit, GTAC is, being the director of GTAC for the last 13 years, is just as guilty of this as everybody else. All of these different data generating facilities usually have their own identifiers that they put on the samples. So tracking something back through 3, 4, 5 different data generating facilities to the original IDs, getting that connected back to the AHR can be a Herculean task, sometimes even for the PI who has the approved protocol. But what if we came up with a way to create a translational identifier of some sort that would get created with that patient sample, and it would, you know how pets today often have a microchip implanted, and the reason for that is that microchip, if the pet ever gets lost, it can be scanned and it can verify, where the pet's home is, right? It's not something you can track directly. It's not a GPS locator. But if you know what the ID is, within that thing, you can actually connect the pet back to the home they belong to. And this is the idea. So the principle here is if we create an identifier that gets connected with these samples before they go into, I apologize, I don't quite have that figured out yet. Before they actually go into our sample collections or the data generating facilities, that identifier could simply go with those samples and track through with them, allowing us to eventually, if you have the appropriate access for clinical information approved protocol, connect back to the EHR. So in GTAC, as we're developing a new LIMS system, we have actually already created a slot for a translational identifier or something like it. So any sample that would come into GTAC can have that identifier associated with it, regardless of whatever identifier we put on it, whatever the initial, the PI's identifier is. This identifier would track through the system and potentially allow us to get back to the EHR if you have the appropriate permissions and protocol to do it. So this is one route in which you're trying to make the connection. In addition to all of that, if you have these sample collections and samples are committed, and especially if you have samples that have some sort of universal consent, as you might imagine, there's the possibility that these sample collections now could also be used to support a lot of human subjects research that allow you to connect back into the EHR. And in fact, Mark Watson is going to be leading something called the BioStore. This is going to be a large sample repository that is actually a piece of what was done with the CTBI. Mark is actually leading this part of it, and these next two slides are Mark's. Where we are basically going to be able to store, in the first version of this, we're going to be able to store about 2 to 4 million vials. This could be DNA, RNA, whatever, stored at -80. And these 200 microliter vials. And it's a dedicated BSL-2 space. It will have a CAP certification and system monitoring, redundancy, et cetera. So obviously, we're going to want investigators to start to use this system for capturing large repositories of samples that we could use for doing prospective research moving forward. Obviously, they own their samples, but if they make those samples available, we're trying to think through what that might, there's a possibility that perhaps at a discounted cost or something like that, if they make the samples available to other investigators to be able to store them here. So there's a number of logistics we're working out with that, and Mark will be the main driver behind that. But the idea is fairly straightforward. A very large biorepository for samples in which we have connections that would eventually allow us to get back to the EHR if we have approved research that allows us to do that. This is a little bit more just kind of a sort of another view of what I referred to in the other slide. So you have your clinical systems where you have your investigator protocols, your consent, the capture of the samples that go into the storage retrieval system. This is a huge room-sized system. It's going to go into the new research, the neuroscience research building over that way a little bit. And basically then these samples could be taken out and products of these samples could be taken out for any of a number of data generating facilities. So those are some of the initial plans about how we are going to target setting up our ability to do this type of connected research moving forward, right? Connecting true translational research along the research axis and then from the research to clinical axis moving forward. So now the other big challenge that I mentioned was the idea of bridging these vast multimodal data, and I'm going to talk a little bit about how we're going to go about that. First, I'm going to show a few slides, conceptual slides of how we're doing it, and I'm going to actually show some examples. So if you kind of take the idea of a human expert system, basically all of us, and you talk about what a human expert system can do and what its pluses and minuses are, at the end of the day, you can kind of break this down in a few things. We know that humans can ingest many types of data and information. We're good at that. We can perform contextual reasoning to identify interesting signal in those data. We're pretty good at that. We can recognize complex patterns and relationships in multimodal data. We're pretty good at that. And we can combine all these things to discover and generate novel hypotheses across those data. So in a lot of ways, the human knowledge engine is really, really good. The one drawback is that we are limited in terms of the quantity and speed at which we do these things by biology, right? Biology can only do some of these things, especially reasoning, so fast. Our circuitry isn't as fast as these new 18 angstrom chips that are coming out on computers, right? It's just physically faster. Plus, the amount of stuff that we can put into a computer in terms of memory and storage is relatively cheap. So there are some ways in which computers do have advantages. The thing is, most computer applications, even things like ChatGPT and some of the LLMs today, don't do a lot of these other things nearly as well as humans yet. So what if we could overcome that? What if we could build a lot of human similar skills into an in silico expert system? And I'm not using this in the terms of the old rule-based expert systems that computer scientists know from years gone by. There may still be expert systems out there that are rules-based. That's why it's in air quotes. It's kind of a philosophical expert system. But this kind of comes back to the CTBI corollary. Can we build a system that is going to allow us to make the human plus the computer much, much more effective than the human alone or the computer alone, and really overcome the limitations that we have within AI systems to do that? So the idea that we came up for doing this and are already kind of employing at a number of levels and have some early data in terms of publications and in research spaces to attack this problem uses this idea. And that is the idea of, and again, I'm not using mixed method in its strict sense here, I'm using it more in as an example, but the idea of mixing different types of artificial intelligence which are actually better at different things, right? Just like people are sometimes better at certain things, different types of artificial intelligence are better at different things too. So model-based, for example, ChatGPT, LLMs, that is model-based AI. For all their sophistication, for all the things that they look like they do, the level at which they're trained, they are a trained system. You might say, well, what's the difference between a trained system and one of these things you're calling a quasi-reasoning system? Well, an example of that would be if I take this wad of paper here and I throw it at Jared, that was a trained response. Jared wasn't sitting there thinking, oh, he's raising his arm, he's got a piece of paper, maybe I should lift my hands up. By the time he's thought of all of that, it's already hit him, right? What he just did was a trained response. We learned it as kids. Something flying at us, we put our hands up and we catch it, right? That is a trained response. That is predominantly what these systems are. It's not a bad thing, but there are certain limitations of trained systems. They can do things very, very well. They can do very rapid evaluation. They can do prediction models. There's a lot of things you can do with these LLMs, but there are some things they don't do so well. For example, they don't actually store true memories of what they know, right? So human beings, while we have trained responses like Jared just showed, another thing I could say, Jared, what's 2 times 2? He just immediately says 4. Doesn't even have to think. It's literally just a trained response. If I say, Jared, what's 72 times 17? He's still thinking. Give him a minute. But that's a reasoned response. He doesn't have that response trained into his system. He has not trained for that question or something in a way that would give him that answer. So humans are kind of unique in that we have both trained responses, and when we don't have a trained response, we can reason the response. If I gave Jared a few seconds, he could come up with the answer because he would use his knowledge of arithmetic and multiplication, and he would simply work the answer out in his head, right? That is more of a reasoned response. It's not an immediate trained answer without thinking, right? Again, both good things, and it's great that humans can do these things. Computer systems are kind of one or the other, right? There's not a lot of reasoning. In fact, there are lots of papers that show ChatGPT can't actually reason. If you give it questions, and multiple papers now that have shown this quite convincingly, if you give it things outside of its training, it'll give you an answer that sounds good, but it won't be right. And how wrong it is kind of depends on how far away the question is from the training. It's not a hallucination, which was what people call these things. It's literally just it didn't have the training to be able to answer that question. In fact, there was a paper that just shows something very similar. It said it gave ChatGPT the question of saying, okay, take a random number between x and y, and then 2 numbers between x and y, and then add those 2 numbers together. Got it wrong, because it literally couldn't reason the response. It did not have a trained solution that could answer that. So that is why we're thinking about merging these two types of artificial intelligence, because there are things that knowledge-based AI can do very, very well that trained models doesn't do so well. You can actually build contextual solutions. You can give it a question, and it can take the context of the question and build the solution based on the data in a near real-time format that answers your question in a contextual format. The solutions are traceable. Everything stored in the system is some type of memory, and it can trace back how it got to the solution. Even though they try to fake that in a lot of the LLMs, they really don't do that very well, okay? And you can even do statistical evaluation because you have other data that you can use that you can find that is nearly similar in context, and you can actually compute response. You can compute a statistical analysis of whether this is a good response or you were likely to find something like that by chance. The last piece of this that we are building is we are going to build this with a very integrated user interface that has a lot of ways to display results that come back from the questions that you ask the system, okay? So, for example, if anybody in the room's used the CompBio platform that we developed in GTAC a few years ago, you know there's a bazillion buttons, bells, and widgets on it, right? In order to get the maps to line up just the way you want and the colors just the way you want and all that kind of stuff. We actually want to build this with a conversational interface that doesn't require a high level of user knowledge of the system. And in fact, we want to build conversational help into the system to be able to use it. And I'm going to show you a little bit about what that's starting to look like in some templates we've already started to build. So, and again, for those who've not really worked with it, you hear about ChatGPT all the time. Some of the folks in the room may have never worked with a knowledge-based AI system. So anybody who has worked with the CompBio platform that we built here, that is actually a knowledge-based artificial intelligence system. It actually stores memories of what it knows. It doesn't train, it simply ingests everything you give it. And it stores it in such a way that it can compute a contextual response to your question, and not quite real time. It's not as fast as a trained system, just like Jared wasn't quite as fast at 72 times 17 as he was at 2 times 2. But it's pretty fast. And you can get answers back in a minute or two as opposed to days or weeks or whatever. And in fact, these are just some of the papers that have been published with the platform that we started building about 6 years ago. And what I can tell you is that this system has been able to identify novel biological findings and generate novel hypotheses that have actually subsequently been tested and validated in the lab afterwards. Okay? And we've even looked at other types of tools like pathway tools and stuff like that. None of the other tools in some of these cases could come up with the solutions that this tool did. So the idea is to integrate a system like this that can do a lot of things that your trained AI, if you built enough different trained models, you might be able to accomplish. But actually take the strengths of both types of systems and put together into a single integrated interface that's transparent to the users. Okay. How about some real examples? It's fine to talk about all this stuff at sort of a hypothetical level. Show us that any of this stuff actually works. So these are small examples, but then I'm going to show you how we're building this at scale. So this particular example, this was a Science Translational Medicine paper that published, I think, back in January. I always want to say December, but I think it was actually January when it finally published of this year. Where Ta-Cheng Liu, myself, and Paul Kelly and a few other colleagues were looking at environmental enteric dysfunction in children. So there were a couple of different studies. These were Gates Foundation funded studies that myself and Ta-Cheng and a few others, collaborators, were on the original papers for, that had looked at EED in children. One cohort was from Zambia. That was Paul Kelly's cohort. And another cohort was from Pakistan, so the Denson group, I think, from Cincinnati Children's did the analysis on that one. The interesting thing is that one of the cohorts from Zambia didn't actually have a control. There was no control cohort for it. So what we were really looking at was the correlation to the primary outcome, which was the length AZ score. Basically, where these children, the problem with EED is it causes stunting. The children simply don't grow. Even when they get proper nutrition, they will put on weight, but they don't grow. And it's been a confusion as to why and what caused that, what's behind it. Well, through a lot of these analyses, we've learned a ton. But there's still work trying to figure out how to overcome that in terms of supplementations, diet, etc. So what we did is we took 2 of the different cohorts because we wanted to find something that was in at least 2 different places and wasn't unique to a particular cohort. And we took the RNA-seq data, and actually the EED from the Pakistan, the SEAM cohort, they actually used children from the US with celiac disease as a control. So not a perfect control, and the other one didn't have a control. So we really wanted to know that whatever we were looking at was showing up strongly in both of those cohorts and wasn't simply an artifact of the controls in one case or the correlation analysis in the other. Well, we did. We actually found a number of biological processes using the AI engine I described on the last slide that actually overlapped within these 2 cohorts quite strongly. And interestingly enough, Ta Chang's lab has been building GI-related animal models for some time now. And one day Ta Chang said, hey, can we compare some of the animal models to the human data to see if any of these might actually be a good match that we might be able to use for some sort of translational type study. So we did. There was Western diet, and there was, I think, a smoking mouse model with an ATG 3L1 or 13L1, 16L1, mutation. There was one in particular, which was a low protein diet, which you might suspect might be a better mimic for these cohorts of kids. So we took the animal models, we did the analysis where we crossed over the animal model biological processes that were basically disrupted in the low protein diet and compared that to the things that were in common between the 2 cohorts of children. And what we discovered was that there were actually 2 components of the biological processes, sorry, this isn't quite working, that were pretty strongly conserved in both the human cohorts as well as we saw dysregulation in the animal model in a very similar way. So Ta Chang's lab used this information and actually looked at 2 different supplements that could be added in order to attack these 2 different pathways as we saw them observed in their dysregulation. And lo and behold, when he put those 2, in this case, NMN supplementation for the NAD dysregulation and cholestyramine for the bile acids, and there's a lot more to that that illustrates why these were solid hypotheses, actually was able to revert pretty much all of the physiological parameters in the low diet, but with these additional components back to or nearly back to the control animals, which were on a 20% diet. And these parts of the world, these children, getting them on a 20% diet consistently is a no-go. Well, these are relatively cheap and easy to integrate supplements. So this gave us the thought that, wow, okay, this is great because yes, we see it in the animal model. You can cure lots of things in a mouse, as we well know. Very few things actually translate. But the reason very things rarely translate is because we don't often look close enough at the animal model to see what it does and does not mimic in the human conditions. And that's exactly what the system is designed to do, and it's designed to do it at scale. So while we've done this on a very small scale here in a very specific study with Ta Chang and Paul Kelly's labs, we're doing this on a much, much larger disease basis scale, which I'll show as we get a little further on. So what about more like the clinical side of this? So another study that we did, and again, Ta Chang's lab is very involved in this as well as Thad and Kelly, and we've also worked with Dermot's group out at Cedars to get these cohorts. So we had 3 different cohorts of adult Crohn's disease. These were collected kind of at different times, or at least were processed at different times over a period of a few years. We had RNA-seq data. These were surgical resections, not biopsies. So the surgical resections, we knew when they were taken, we know why they were taken, and Ta Chang's lab was very careful to take regions away from the inflamed necrotic regions at the site for which the resection was being done. So we're actually seeing not a bunch of inflammation, but we're actually seeing what was going on in the intestinal epithelium, the crypts, et cetera, which is very hard to do if you're just getting an inflamed region. Everything's just overwhelmed by the inflammation. We took these into the system, and with a new aspect of the system that Chad Storer from our group developed last spring, we were able to take these cohorts and we were able to break them into 5 distinct molecular subtypes using the AI engine again. And when we did that, after we did that, so these were all just based off the RNA-seq data and knowing where these came from and knowing they were kind of lined up with the surgical resections. We then mapped histological, phenotypic, and outcome data onto these 5, right? Looking to see if there was anything that these 5 molecular subtypes were predicting. Because if you've got molecular subtypes, but it doesn't predict anything useful, then who cares, right? Well, it turns out that they may be predicting something incredibly useful. So the most important of all of these is the outcome data. Basically, how rapidly these people progress in their disease after surgery. So basically, this is looking at the duration in months from the time of the surgery to the time of recurrence. And what you may notice right away up here, what we call C2 or cohort 2, is that the vast majority of that cohort had an extremely rapid recurrence after surgery. If you look over at 5, hardly any of these subjects had a really rapid recurrence. In fact, most of them had relatively long recurrence durations. It turns out that the C3 and the C4 actually are very similar to C2 and C5, going in the opposite directions. They separated somewhat because of actually the BMI of these subjects. So that separated their biology just a little bit so that when we did the clustering, they came out as separate. But you can actually see there's a very large number of people with a very short duration here, and again, very few. Most of these are long duration individuals, just like cluster 5. And in fact, we're now digging into this and we're finding molecular mechanisms and components that we think are the actual drivers of this. Now, from the clinical side, the question you should probably be asking is, okay, these might be, C5 and C2 might be a different molecular subtype, might be differences in etiologies, lots of different things might cause that. But within C2, if I can get the pointer right, there's a relatively small group that had a relatively long duration to recurrence. Why? What if our system had all the electronic health record data of these people in the background, and we could simply ask the system, compare the short duration of C2 to the long duration of C2, and tell me if you're seeing any differences in their treatments, in their comorbidities, and other drugs that they may be on for other unrelated comorbidities. And I suspect we would find, in fact, we've even got a thought about what might be driving that. We don't know yet until we would be able to do this analysis. But this is exactly what we want to build into the system. Because in this case, you don't have to return any PHI. The computer system can know the PHI as long as it knows what it can and can't tell the person who doesn't have a sufficient protocol with permissions to see that PHI. So we could actually do within the system the analysis I just described. Take the group 1 and the group 2 within cohort 2, go off, pull in all their EHR data, and see if you can identify anything distinctly different within those 2 subgroups within that molecular subtype. This is exactly what we're trying to build into the system and make this available to everybody within the institution to do this kind of research. And that's where the data bridging I talked about before is so important. Okay. So those were small scale examples. We are now doing this on a very large scale. So we picked Alzheimer's just because as the first place we were going to do this at scale because the amount of data are vast. And basically if we get the system working there, everything else is frankly relatively simple by comparison just because of the amount of data that we have to process. So we have, in this case, we're assembling very large collections of human omics data, hundreds to thousands of samples in patients. In many cases, multiple tissues and omics types. I didn't mention the Comp biosystem can handle multi-omic data. If you give it metabolite data, protein data, gene expression data, can handle all of those. And basically, it will do the molecular subtyping and give you the ability to connect the phenotypes. We're also making extensive collections of related animal models. So for example, in the Alzheimer's space, we're in the process of collecting about 2,000 to 3,000 subjects worth of data across multiple cohorts. We already have the ROSMAP cohort mostly processed. We have a few others that we're bringing in. We have about 70 different neurodegenerative animal models. And basically what you see over here is what we're doing. We're taking all of the animal model profiles, all of the human subject data, subtyping the human subject data, and then we are basically building a map across the animal models and subtypes by biological pathways and processes, as well as entities. And basically giving people the ability to ask a lot of questions that you might imagine. So for example, Fuhai Li's group, Fuhai's a collaborator in I2DB, and his group has done a lot of work with drug repurposing, where they've built a lot of machine learning models to look at drug repurposing. And what we are going to do is we're actually going to take the models and the profiles they have built for their drug repurposing using machine learning techniques, and we're going to overlap those with our disease models with our animal model and human model data, looking for translational pathways across not only which animal models might you want to test those particular repurposed drugs in, but which human cohorts would you probably want to take it into first, okay? So I've got a hypothesis, I've got a target, I've got a drug, I've got a model, say I've got an organoid model, something like that, and I've got enough of a profile or information around it that I want to compare that in a translational fashion across this to figure out the best chance of success if I start testing this in a translational mode. And so we're doing this now. We've collected, as I said, we've collected a lot for AD. We're beginning an ALS collection shortly, probably about 2,000 subjects worth of data there, Parkinson's to follow that, and then a number of different disease areas that are going to come in right behind that. And then basically take this all the way to the point where we have it right up to the spot of that EHR connectivity. As you might imagine, that's only going to work where we have people from a WashU system where we can connect back to the EHR, but as we move forward, a lot of the data we generate here are for those subjects, right? So this should become much, much more useful in terms of that last step of connection as we have more and more subjects worth of data that we can track through the system that way. So this is just to show that we are also working on an integrated use environment. So what you kind of see over here are the first pinnings of the model and disease analysis interface, but you may notice that there is basically a chat bot built in with it. And in fact, we're doing this with CompBio and a number of other tools as well, where the user will have access to both the knowledge base AI and the modeling base. In this case, you come up with an idea. You see some connection in a CompBio map. Oh, peroxisomes and beta oxidation. How are they connected? You simply ask the question in the chatbot window and potentially, hopefully, get back a right answer. But the idea is to be idea generating as well as to put the help for the system into that same window. So it can basically help give you ideas about how to use the system effectively and how to get to hypotheses and or generate ideas effectively. And we're going to be doing this at every level of the system, as well as integrating other models into the system, like I mentioned, in the case where we're going to try to integrate what Fuhai is doing with what we're doing. So this gives you a little bit of a flavor. Just a quick summary. I think I've kind of mentioned all this. Secure sample and data bridges are the first step. Highly advanced computational framework to support it. We're going to leverage these systems to really enable translational research at a previously unrecognized scale and speed. We're going to create an integrated environment to make this accessible to WashU, all WashU users, and rapidly advance discovery and adoption of precision medicine initiatives, which frankly have been rather slow in uptake. A lot of people in the acknowledgments. David Perlmutter for getting us to the point where we have the funds to do this. Jeff Milbrandt and Philip Payne, who are the respective directors of the MGI and I2DB. Ting, who's been a huge help in faculty recruitment and Department of Genetics. You've already heard a lot about a lot of the key collaborators. Albert Lai's team is a huge, huge boost for what we're doing because they've built the data lake around the clinical data. Our core team, a lot of folks from GTAC, Kim, Dan, Jesse, all of the folks from MGI and I2DB business administration teams have been a huge help in getting all this off the ground. A lot more I probably can't even cover. Quick shameless plug, there is going to be a proteomics symposium. It is very tied into what we're talking about, where it's very focused around disease research and biomarker discovery. This is going to be on October 17th. We have Joe Koresh, who's like one of the most cited authors in the country. He has over 350,000 citations. He has one paper with 70,000 citations. And he does an enormous amount of high-plex proteomic work. Ali Havahari from WashU and Carlos Cruchaga, who I think a lot of people probably know, has done a lot of work in the AD space with multiomics. So on that note, I will stop, and if there are any questions, I'm happy to answer them. There's probably still some refreshments outside, so we've got a little time to chat out there too if anybody wants, but otherwise I'm happy to take any questions.